Hello students, today we are going to start with the respiration in plants. So firstly, you have to understand this thing that in plants there are no specialized organs for the respiration process, right? Respiration is actually the breakdown of two carbon-carbon bonds by the process of oxidation, okay? Now the carbohydrates, they get oxidized to release energy. On the other hand, if we talk about proteins, fats and organic acids, those can be used as the respiratory substrates, right? Now ATP, we know ATP, that is adenosine triphosphate, is the energy currency of the cell. Energy currency of the cell, ATP, is produced by the reaction called glycolysis that we are going to discuss. ATP will be actually firstly used in the process and then it will be produced. Okay, it is produced with ADP, adenosine diphosphate is used, and adenosine triphosphate will be produced in the second phase, in the payoff phase. Okay, so when we are going to start the reaction glycolysis in plants by the process of photosynthesis, there are four products that are normally produced. Those are fructose, starch, glucose or sucrose. If it is glucose, glucose can immediately start the process of glycolysis. On the other hand, if it is sucrose or starch, it first needs to be converted to glucose. If it is sucrose, it will be converted to glucose with the help of enzyme invertase. And if it is starch, it will be converted to glucose with the help of enzyme amylase maltase. Okay, once it has converted to glucose, glucose will then use ATP, adenosine triphosphate, 3 phosphates, and it will form ADP, adenosine diphosphate, 2 phosphates. So means one phosphate has been taken up from ATP. That one phosphate has attached at the 6th position of glucose, forming glucose 6 phosphate with the help of Mg2 positive ion and the enzyme hexokinase. Okay. Now 6th position phosphate has attached. Now this glucose 6 phosphate will change its shape. From glucose it will form to fructose, that is actually an isomer of glucose. So glucose 6 phosphate will form fructose 6 phosphate with the help of enzyme phosphohexose isomerase. Isomers, they both are isomers of each other. The formula, if you see, is the same. The number of carbon, number of hydrogen, number of oxygen is the same. Just the shape, the structure of the compound is different. That is why they are the isomers. So, phosphohexose isomerase will be used in the presence of Mg2 positive ion. Now, till here we have done, right? In case fructose is the product of photosynthesis, fructose will directly get converted to fructose 6 phosphate with the help of ATP, that is converted to ADP, 1 phosphate used, and that 1 phosphate has attached to the 6th position of this fructose, right? And the enzyme used here will be fructokinase. Kinase, what is kinase? Kinase is an enzyme wherever the ligation is being done, the pasting is being done, kinase enzyme will be used. Okay. So fructokinase means 1 phosphate has attached to 6th position forming fructose 6 phosphate. Here also hexokinase. Okay. Then after fructose 6 phosphate, we will take another ATP and form ADP, again using 1 more phosphate. 1 more phosphate we will take, and that will get attached to the first position of fructose 1, forming fructose 1,6 biphosphate. 1 phosphate and by 2 phosphates. 6th position already had the phosphate group. Now the first position also has the phosphate group, and this happened in the presence of enzyme Mg2 positive and the enzyme phosphofructokinase. Again, kinase, just mark these things to remember the name of the enzyme, kinase. Everywhere the phosphate has attached kinase enzyme is used. Okay. Now after fructose 1,6 biphosphate, in the presence of enzyme aldolase, triose phosphate will be formed. Triose phosphate is the name given to both of these compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of these are interconvertible. Okay, any one of these is formed and two molecules will be formed. Okay, interconvertible, so they will be two will be formed and then one of them will convert to the second one. Okay, so this is actually the preparatory phase. Okay, this is the preparatory phase. In this we have used ATP and ADP is released, means 2-phosphate we have taken up from this, right? Now if you want to just look into this into more detail, I am going to explain you once again this reaction but with the compound, so that it becomes easier for you to understand कहाँ से phosphate लगा और कहाँ से वो remove हुआ. Okay. Now I am going to draw this all of these compounds, structures, formulas. Okay. So glucose, you know, this is the glucose molecule. One carbon has CH2OH. Okay. Now this is only where our phosphate group is going to attach. This sixth position. This is the sixth position. You can count. O is here. This is the first, second, third. I'll just write it. This is the first, second, third, fourth, fifth, and this is the sixth carbon. On sixth carbon, this O is there. With O, this phosphate group will be attached. Phosphate always, oxygen always has double bonds. Okay, either it is with one molecule or with the with some two compounds, and phosphate has 5 bonds. Okay, so we have to keep this thing in mind when we are doing when we are drawing the structures. So this is the glucose. Now glucose 6-phosphate will be this. I am not going to write OH again and again. Okay, so CH2O. This is as such. Now we will remove H. H will be removed and instead there will be P. Phosphate has 5 bonds, so one is with O, one double bond with O. This is 3 bonds. Then 2 with O negative. Okay, we can also make it HOH. Okay, but here we are going to take 2 negative. Okay, this is glucose 6-phosphate. Sixth position of carbon has the phosphate group. Then fructose. Now here it is a hexagon. Fructose will be a pentagon. It will be a pentagon. Okay. Now here CH2O is on the 6th position. This is on the 6th position. O, P, double bond O, O negative, O negative. Right. On first position also same way CH2 is here. Okay. So this is CH2OH. Now you count. This is the first carbon, second, third, fourth, fifth, and this is the sixth. First, second, third, fourth, fifth, sixth. Right? The sixth carbon already has this phosphate group on the sixth position. Now first carbon will also have this phosphate, this one. Okay. So this will be the pentagon. Then CH2 O P double bond O negative, O negative. Right? This is the sixth carbon, and on the first carbon CH2 O P double bond O negative, O negative. How many bonds? 1, 2, 3, 4, 5. Right? 5 bonds have been made. Now this is fructose 1,6 biphosphate. Now this we have made this fructose 1,6 biphosphate. Now this will open up to make dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which are interconvertible and will form the same compound. Okay, but two compounds will be formed. Two of them will be formed. Any of these is formed. Okay, these are interconvertible with the help of enzyme triose phosphate isomerase. Okay, so now you see from here it has been cut off, and the this pentagon has opened up. Okay, and the chains are formed. Okay, so if we talk about dihydroxyacetone phosphate, one phosphate group has come here. Okay, and CH2 and double bond O. This O has this O is in both. See this one and this. Okay, what has happened now? When there is O is joined with the chain with only one compound, it has to form two bonds, right? So the double bond will be formed with this one also and with this one also. Okay, so this O, then there will be OH, one CH2 and then OH, again CH2, and then there is the this phosphate group. Same. Okay. Similarly, in this one, phosphate group, and then this CH2, OH, and this double bond O. Okay. So both of these open up. Okay, and the further the payoff phase starts. Till here, it is the preparatory phase. Preparatory phase is the initiation of the glycolysis. Here we have used 2 ATP to form 2 ADP, means 2 phosphate molecules have been attached to the compound. 2 phosphate molecules we have taken up. Okay, so this is the net reaction of the preparatory phase. Okay, for the payoff phase, stay connected because the next video is going to be for the payoff phase. Thank you.